Okay, this is our second lecture. It's not going to be as long as yesterday's lecture, but we're going to do the chapter that deals with the other prokaryotic domain, which is the archaea. And hold on a second. I'll get the chapter. I need to find the chapter. Just hold on a second. I will have. Okay, sorry for this. I need to just get the chapter ready. Now this. Okay, yes. So here we go. Okay, so this chapter discusses archaeal cell structure. It is a shorter and lighter chapter compared to the bacterial structure. Recently, in this book, they decided to give a separate chapter to archaea because they are quite different from bacterial cell, and the old system of grouping them together in order to just discuss the prokaryotic cell kind of made it harder maybe for people to recognize how different these cells are. It's true that they are prokaryotic cells, so they don't have a nucleus, but at the same time, they are belonging to a separate domain compared to the bacteria. And so they do have very many important differences. And so in this chapter, we're going to learn and look at some of these differences. So they are prokaryotic cells, and so they don't have a nucleus. Their chromosome is most of the time circular, like the bacteria. Sometimes there are linear chromosomes, so the chromosome has a beginning and an end, is not a circle. The thing with archaea, which makes them an interesting, very interesting group, is that they are kind of in between the bacteria and the eukarya. In the past, it was thought of archaea as very old. The name archaea means very old. And it looked as if they were the precursors, that they were the type of cells before bacteria. But now this idea has been revised. And if there is a common ancestor, the common ancestor split and gave the bacteria on one side, the archaea on the other, and then from the archaea we get the eukaryote. So the name archaea and this idea that it means they're older than bacteria, it's not correct. If I told you yesterday that we have isolated a very large number of bacterial species, and we have 40 different phyla for bacteria, the same is not true for archaea. We know much less when it comes to archaea. They're a little bit harder to isolate because several species of archaea, you could say maybe the majority or a very large number of archaeal species actually are adapted for living in extreme environment. And in order to isolate, grow, and study these cells in the laboratory, it is necessary to re-establish those conditions. And this can be quite difficult. It's difficult to find the right media for growing them, the right temperature, the right pH. And so there is less, we know less of archaea compared to bacteria. It is also true that until now, there is no disease, human disease, or even plant disease, or animal disease that has been identified as caused by an archaea. And so also there is less focus on understanding and learning bacterial biology. It's not that is not important or that is not interesting, but you know how science works, and sometimes science requires money, and there is more money for research in fields that affect us directly rather than pure biological and environmental studies. Anyways, when we look at the archaea, we find that we can find a lot that is common to bacteria and a lot that is different. When it comes to things that are common between bacteria and archaea, we see that a lot of the genes for metabolic activity are similar or in some cases identical. So you have that flexibility that we kind of mentioned yesterday, with the fact that the cell is not constrained by having organelles, which is also a characteristic of archaea. So genes for metabolism are in common with bacteria, but when we look at other things, like for instance, the ribosomal RNA, the genes that codify for the ribosomal RNA, we find that archaea have unique and different sequences which separate them from the archaea. They are also, till now, the only type of organism that is capable of reducing carbon dioxide to methane gas, so they are capable of methanogenesis. They are very different from one another. As with the bacteria, they're different in terms of morphology, shape, physiology, reproduction, and ecology. As I said before, as a general statement, it's not that all of them, but most of them are very well adapted to live in environments which are classified as extreme environment. What makes an environment an extreme environment? It is an environment where other cells, other organisms will die, basically. Yeah, so they are good at growing in anaerobic conditions, so without oxygen, hypersaline, very high concentration of oxygen, so high hypertonic environment. pH extremes, either very low or high pH, and high temperature. So they are well adapted for those conditions. This table kind of summarizes a comparison of bacteria and archaeal cells. When we look at these major components of the cell, we see that there are some important differences. For instance, the plasma membrane, the lipids that make up the plasma membrane of archaea are quite different chemically compared to the ones of bacteria. We're going to talk about these a little bit more in detail later. Also the cell wall, you remember we said yesterday, bacteria have peptidoglycan, archaea don't have peptidoglycan, there is no peptidoglycan. There are inclusions like bacteria, the ribosome size is the same, 70S. The chromosome structure is circular, and as I said, archaea also are mostly circular. I think there is now, they isolated now one that has linear DNA, but I'm not sure about that. They have plasmids, both of them, and external structure, flagella is common both in archaea and bacteria. Fimbriae and pili are in common, but archaea have also other structure like hami, for instance, and cannulae, which are specific to archaea. The capsule or slime layer are common in bacteria, but not so common in the archaea that have been so far isolated. So like we did with the bacteria, we look at archaea shape, arrangement, and size. Again, the coccus and the rod are the most common shape. They are the most easy shapes in terms of ratio between volume and surface. But there are other shapes. Actually, the first archaea ever to be identified were isolated in the Sinai Desert, Ficina, in Egypt. And they had a square shape, which is not something that is quite common in nature. Yeah. So far, we haven't isolated spirochetes or mycelia. They can be branched and they can be flat. Yeah, flat shapes like the square shape. The size is similar to the one that we said. The average size is similar to the one that we discussed for bacteria. 1 to 2 by 1 to 5 micrometers for rods and 1 to 3 micrometers in diameter for cocci. There are very tiny ones, 0.2 to 0.4 micrometers, so 200 nanometers in diameter. And we have been able to isolate a multicellular form of archaea, which is an exception, that can reach 30 millimeters in length, so 3 centimeters. So that's quite big for a microorganism. Yeah, and here there are some pictures showing you coccus archaea. This is a branched one, Thermoproteus. You see how it has this Y shape, yeah? And in this picture here on the right, there is an archaea. You see here the filamentous archaeon, which is covered by bacteria. Yeah. So if we look at the envelope, like we did yesterday, we can start by looking at the plasma membrane and then the cell wall and the external layer. Sometimes the cell wall is made only of protein, the S layer. You remember how we talked about the S layer, proteins that are good at self-assembly? So outside the plasma membrane, in some species, there is only a cell layer. Some don't have a cell wall. Again, these are the ones that are found living inside other cells, and the capsule and slime are rare. So let's start looking at the membranes. Membranes of archaea are very interesting. There is some archaeal cells that have membranes which are very similar to the one of bacteria and eukarya with the bilayer of phospholipids. But even if it is a bilayer of phospholipids, the lipids, so the fats, which, if you remember, are formed by glycerol and fatty acids, when the glycerol binds with the fatty acid and there is the condensation reaction that removes a molecule of water, instead of forming a ester bond, bacterial fats are bound through ether bond. And this is true for all the fats of archaea. And so this is a major biochemical difference between archaea and bacteria and eukarya. Both bacteria and eukarya, they have ester bond. With archaea, we never have ester bond, we have ether bond. It also, you can have isoprene units, which is 5-carbon branched lipids. And as I said, ether linkages rather than ester linkages to glycerol. Some of them are phospholipids. Some of them have sugars instead of the phosphate to make the amphipathic molecule. And some of them, quite interestingly, instead of making a bilayer, you see here, you have the 2 layers of fat, they make a monolayer. So the 2 hydrophilic heads are joined by a continuous fatty acid string. Yeah. So the membrane lipids are different from bacteria and eukarya because they have branched chain hydrocarbons attached to the glycerol using ether linkages. They are polar phospholipids, but you have also sulfolipids. So you have sulfur instead of phosphate. You have glycolipids, sugars instead of phosphate, and some other unique lipids found in archaeal membrane. And this figure here shows sorry, there is this little fly that is shows the difference between bacterial membrane here on the right and archaeal membrane on the left. First of all, let's look at the linkage. You see here you have the glycerol and the fatty acids attached to the glycerol. With the archaea, you have an ether linkage. With the bacteria, you have an ester linkage. So there is a carbonyl group here with a double bond with the oxygen that is not present here. Yeah. You can have isoprene derived hydrocarbons in archaeal membrane, and the head group is you know, the charged group, but the charged group can be something different than phosphate. So with bacteria, we have the bilayers. It's always a bilayer, yeah. With archaea, we have bilayer. It could be something that is very, very similar to the bacteria, like here number 3, you see it's very similar, or it could be monolayers. And you see here the monolayers that can be quite straight molecules or containing rings, which make it a bit more flexible. Of course, you kind of probably already made the connection that a monolayer compared to a bilayer is a much more rigid structure. Now, if you compare two different types of monolayer, like this one that has a straight molecule and this one that has the rings that kind of stretch and make the molecule wider, and this one, number 2, is less rigid compared to number 1. But both of them, 1 and 2, are much more rigid than 3, 4, 5, and 6. Yeah, so this type of adaptation to the membrane, having monolayers, is strongly associated with adaptation to living in extreme conditions, especially very high temperatures. You remember how we said the temperature affects the fluidity of the membrane, and hopanoids in bacterial cells, cholesterol in eukaryotic cells, work with making harder for the temperature to change the fluidity of the membrane. And if you change the fluidity, you change the permeability. But when you go to very high temperature, above 60 and 70 degrees, which is where some of these archaeal species live all the time, that is not enough. You need to have more systems in place in order to stop your cell to start leaking cytoplasm outside. And these monolayers are one of the mechanisms which help with keeping the fluidity of the membrane correct at high temperature, okay? So when they are like this one, the monolayer, like for instance, number 1, they're called diglycerol because you have 2 glycerols here, one here, one here. Tetraethers. Tetra because it's 4, yeah, 4 ether groups. There is one here, one here, one here, one here. So that is the major difference between the bacteria, the eukarya, and the archaea. It puts the archaea in a group completely separate. No ester bond and monolayer in the membrane. The plasma membrane can have S layer attached to it. It can have part of the cell wall made using a polymer, which is similar to peptidoglycan, but is not peptidoglycan. This is called pseudomurane. Remember yesterday we said that peptidoglycan used to be called murane. This looks like murane, pseudo, but is not murane, so pseudomurane. Or you can have polysaccharide, you can have proteins, glycoproteins. Generally speaking, until now, only one species, one genus, Igniococcus, has been isolated with an outer membrane, so with the architecture of the cell wall that is more similar to the one of the gram-negative, having an extra membrane outside. The membrane is a barrier, it's a semi-permeable barrier, and it allows for material to move across the membrane. It is also, like with bacteria, the place for some metabolism. And when it comes to obtaining nutrients, archaea use the membrane in a way that is very similar to the one that we saw with bacteria. You have facilitated diffusion, you have active transport, primary and secondary, you have the phosphenol pyruvate sugar phosphotransferase system, PTS, the group translocation that we discussed with bacteria. So, you know, the transport system doesn't seem to be very different, but the cell wall are different. Yeah. So the most common type of cell wall is the S layer. So there is no sugar, or there is sugar because the S layer could be glycoprotein, but there is no polysaccharide in the structure. You can have polysaccharides attached to the protein. Some of them have protein after this S layer as well. You might have a layer of pseudomurine between the S layer and the membrane, or you might have pseudomurine as the most outer layer, so in a way making it similar to the gram-positive cell wall. This picture here shows the different combinations. You remember yesterday with bacteria, we said either one or the other type. There is nothing else. The archaea, we have very many different ways of making the cell wall. This is the pseudomurine and acetylaziminuronic acid and L-acetylglucosamine. So the disaccharide is different. You still have the chain of amino acid, but you don't have the interpeptide bridge with the glycines. So it's different structure, pseudomurine compared to peptidoglycan. And this scheme here shows different types of cell wall. You have type A, where you have the cytoplasm, the plasma membrane, and then the S layer, so very easy. Then there is B, plasma membrane, S layer, and then another layer of protein outside. C, which is S layer, and methanochondroitin. Chondroitin, maybe you know it from other courses, is found in cartilage in humans and in other animals. Methanochondroitin is a sugar and protein-based substance containing methane. This is found in the archaea that produce methane as part of their metabolism. Yeah. Then you have, you can have plasma membrane, pseudomurine, and S layer. So this, in a way, is the one that is most similar to the gram-positive cell wall. You can have a plasma membrane and sugars, polysaccharides. You can have plasma membrane and glycocalyx. So this is not really a cell wall. Or you can have a plasma membrane, outer membrane, and an intermembrane compartment that doesn't have any pseudomurine. Okay, so you see how we have a much more varied combination of possible cell walls architectures. Okay, I need to, okay. So that is the cell wall. The cytoplasm, very similar. There is no membrane enclosed organelles, inclusion bodies, ribosome, nucleoid region, but there are some differences when it comes to the ribosomes. Even though the bacteria and the archaeal ribosome have the same density, there are some differences. When you look at the large subunit of the ribosome in archaea, it contains the ribosomal RNA. Ribosomal RNA that can be similar to the one of our bacteria, but also it has a 5' 5.8S ribosomal RNA, which is also found in eukaryotic ribosomes. So it's like the ribosome is a bit of a mix between the bacterial ribosome and the eukaryotic ribosome. Also, the proteins that make up the ribosome are a little bit different. The proteins are a little more similar to the ones of eukaryotic cell compared to the ones of a prokaryotic cell, and there are also some proteins that are found only in archaea. The nucleoid is where the chromosome is located, single circular chromosome with associated proteins. Some archaea look as if they might have more than one chromosome. You have supercoiling in order to condense the DNA and fit it inside the cell with nucleoid associated proteins. And here, in this instance, we find that some of these proteins are actually histones, like the proteins that are used by eukaryotic cell, and these help in folding and chromosome condensation. External structure, we have the pili, which are similar to the pili of archaea, not yet 100% studied, but similar to the ones of bacteria. Pilin protein is the protein that makes up the pili. They're probably, the function is probably for attachment. There is also cannulae. Cannulae are specific to archaea. They're not found in bacteria or in any other type of microorganism. Cannulae means little straws, yeah? And they are like tube structures, which can be found on the surface of the thermophilic archaea in the genus Pyrodictium. You see them here. You see these little sticking kind of structures. They're empty inside. They're like a straw, you know, like a massasa, yeah? The function of this is unknown, but it might be making networks of cells that stay together and form a community of daughter cells. Then there is Hami. Hami, here in this micrograph here, you can see this curved structure. Hami comes from Latin, which is for the hook that you use for fishing, yeah, because they have this hook shape, yeah, probably involved with adherence and attachment to surface. And then we have the flagellum. The flagellum is made not only of one protein. In bacteria, we have only flagellin making the flagellum. Here there is more than one type of flagellin. It's thinner. It's not hollow, so the filament is not an empty tube, but is full of, it's a solid structure. It is difficult to distinguish between the hook and the basal body. And I don't know if you remember yesterday when we were discussing the flagellum of bacteria, we said that the flagellin is made in the ribosome and then it goes out through the hollow tube, and it adds to the length of the flagellum from the tip. Here, because this is not empty, so you can't push the protein through, it adds at the end, yeah, and so the growth is adding at the end and making it longer. There is different types of flagellin protein, 1, 2, and 3, flagellin 1, flagellin 2, flagellin 3, so it's not the same as the bacteria. So, well, basically, this is the end of this chapter. As you can see, it's not much. I don't want, because there is nobody today connected, I don't want to make this a long lecture, but I want to finish this other chapter, so that tomorrow, when we have our lecture, we can go and move on and start looking at some bacterial growth and how the cells grow and how they divide and so on. Remember that the class tomorrow is not at 4, but it is at 5, because I have seminars. I will send a message anyways on ITC. Make sure that everybody knows that the class is at 5. Okay, so let's keep it short and nice for today, and I will see you tomorrow. Goodbye.